Hello everyone. Now in our previous video on enzymes, I had finished up with the factors affecting the activity of the enzymes. Along with I discussed the kinetics of enzymes. So I'll just give you the gist of what all we discussed in our previous video. We had discussed about the kinetics of enzyme, in which one thing which you have to actually learn by heart is that Michaelis and Menten were the two scientists who gave that kinetics, and according to that the equation is V naught is equal to Vmax S over Km plus S. Now why in this video also I am just telling you to go through this, because kinetics plays a very important role in actually giving the activity of the enzyme. And yesterday also I told you ki what are the enzymes? They are the biocatalysts which are increasing the rate of a biochemical reaction. So always keep this thing in mind that this Km plays a very important role. Okay, whether it is hexokinase, whether it is glucokinase, you name any enzyme, every enzyme will have its specific or own Km. Okay, and I told you one more thing, that this Km is inversely proportional to the affinity of the enzyme with the substrate. And we all know that inversely proportional relationship means if an enzyme has low Km, automatically affinity would be high. And if we talk about more Km, high Km, the affinity needs to be less. Okay, an example. Again, we are going to discuss these examples in detail also, but here just for your knowledge, I can say that low Km, if we talk about, the most ideal example is hexokinase. And high Km example is glucokinase. And in general, if we talk about any ideal substrate, just keep this thing in mind. All ideal substrates will have a low Km. They need to have maximum affinity. So I can just write it here that all the ideal substrates. Then we discussed about the reciprocal of Michaelis-Menten equation, which is known as Lineweaver-Burki's plot. And what is the significance behind this is because Lineweaver tried to, give a relation between the slope and the intercept, and they actually reciprocated this equation. Okay, and it comes out to be a straight line like this. So if you want any detail, you can just go to my previous video. Okay, this becomes the intercept and this becomes the slope. So I can say that slope is Km over Vmax and intercept is 1 over Vmax. And this is going to play a very important role when we are going to study the inhibition. So this is all about the kinetics. Now after this we are going to deal with a very important topic which actually correctly coincides with the kinetics, that is enzyme inhibition. Now see enzyme inhibition, because all the biochemical reactions, they are actually governing the rate of the rate by lowering down the activation energy as previously discussed. So if we talk about enzyme inhibition, there are so many different ways of classifying that they are into what classes. But if we talk in general, I can simply say that enzyme inhibition can be reversible, can be irreversible, right, and suicide inhibition, which is altogether a different type of inhibition, and the most important inhibition is allosteric, which is actually known as modulation. But yes, if we see the definition of allosteric enzymes, we can say that they inhibit also in a way they are modulating. So we can use the term allosteric modulation or we can use the term allosteric inhibition. So these are the four major type of inhibition. Now first of all, if we talk about reversible, as the name suggests, it can be reversed back. Okay, so if we talk about reversible, the most important type of reversible inhibition is competitive inhibition. Competitive inhibition. Now, this is strictly reversible. A very important point about this. now see, again it is clear from the word itself there is a competition. Competition किसमें? Between the inhibitor and the substrate. Okay. So if we have to write the equation, it's very clear. This is the enzyme. In normal conditions, it is going to combine with the substrate, forming the enzyme substrate complex as we have discussed, and it will be dissociated to form the enzyme and the product. But now there is a competition. Okay, competition between the substrate and the inhibitor. Why there is a competition? Because inhibitor is resembling the substrate in structure. So enzyme by mistake, instead of binding with the substrate, binds with the inhibitor. So obviously the game will change. So we can say that enzyme inhibitor complex will be produced. So obviously the desired product will not be there. Okay. So I can simply say that what is there? There is a competition between the substrate and the inhibitor. Now the question is, if there is a competition, what will happen to the kinetics of the enzyme? That is what is our main target, because we have to see now how enzymes are combining with the substrate, or when an inhibitor is binding with the enzyme, what role it plays. So what happens here is Vmax, because there are two main parameters, Km and Vmax. So always remember, in competitive inhibition, Km will always increase. Why it will increase? Because the affinity of the enzyme with the substrate will decrease. Okay. So I can say Km increases, so the affinity will decrease. And on the other hand, Vmax will remain unchanged. Now the question is, why Vmax is unchanged? Plain and simple. Whether the substrate is binding or whether the inhibitor is binding, both are resembling each other in structure. So there can be no change in Vmax. This is very, very important. So always remember, in competitive inhibition, Km is always going to increase, whereas Vmax will be unchanged, or there will not be any change. Right. And if we have to plot a graph, if I talk about Michaelis-Menten graph, we can simply say that it will attain like this, and in the presence of the inhibitor, it will be like, or you can say that here Vmax will be same, but if we talk about the Km, we can see that it is like this. This becomes the Km. So we can say Km increases, whereas Vmax same. So this is as per Michaelis-Menten. We can say that here we simply velocity will be there, and here we will say that substrate concentration will be there. Now if we have to change this graph, or if we have to extrapolate this graph, or you can say as given by Michaelis and Menten. After that, if we talk about the line-Weaver-Burkes plot, here what will happen? Again, the parameters are going to remain the same, but Km and Vmax, if we talk about, it will be like this. Like this. So this becomes the intercept, or you can say Vmax will be, because it is the intercept, it will be same, whereas again we can say that 1 upon Km, because this is this side. So we can say that this is actually a X-shaped graph. So you can say that if we talk about line-Weaver-Burkes plot, it becomes X-shaped. So suppose if it is asked in the exam, and this type of, because this is generally asked as an IBQ image-based question. So if this image is asked, simply by seeing X-shaped, it has to be competitive inhibition of line-Weaver-Burkes plot. And here you can simply say that this is Michaelis-Menten, right? So this is competitive inhibition. Now what is the importance and what are the examples of competitive inhibition? So if we talk about the examples, the most common example of competitive inhibition is malonate. is a inhibitor of succinate and the enzyme is succinate dehydrogenase. Now this is a reaction of TCA cycle. We all are familiar with this reaction. So basically what is happening is, we know that succinic acid or succinate is converted to fumarate and the enzyme is SDH. Now inhibitor is what here? Inhibitor is malonate. So malonate is going to compete with SDH. So when malonate is there, no fumarate will be there. So this is one very important example. Second important example I can quote is methanol and ethanol, because again if we talk about, there was a huge tragedy, there is a clinical application also. If a person is so much into drinking, or suppose if a person ingests alcohol, methyl alcohol, we know methyl alcohol is highly dangerous, immediately ethyl alcohol should be given as an antidote. So I can simply say that methanol or ethanol. In huge tragedy, the same thing happened. Because what happens is, alcohol metabolism is altogether a different metabolism which we are going to cover in the next topic. But here I just want to tell you this, ki again this is a kind of competitive inhibition. So ethyl alcohol is generally given as an antidote because methanol is actually poisonous. Because methanol can be easily converted into formaldehyde or formic acid. So formic acid is highly poisonous. Okay. So these are the two examples, very ideal examples. Now there is a therapeutic use also of competitive inhibition. What is that therapeutic use? Very important is, which is actually asked in the exam. Therapeutic use is we know about the statins. Statins is a class of drugs. First example, statins. Statins are a class of drugs which are given to those patients who have high level of cholesterol, who are suffering from hypercholesterolemia. So obviously when statins are given, what should be their mode of action? They should decrease the synthesis of cholesterol, and that is what exactly happens. How? Because statins are the competitive inhibitors of HMG-CoA reductase. Very important, right? And what is HMG-CoA reductase? It is a rate limiting enzyme of cholesterol biosynthesis. And if we look at the reaction, what is actually happening? Because we know that cholesterol, no doubt we take cholesterol in the diet also, and cholesterol is synthesized in the body also by a very important de novo pathway, and precursor of cholesterol is acetyl coenzyme A. Now what is happening here? We can simply say that this HMG-CoA is converted into, HMG stands for beta hydroxy beta methyl gluteryl CoA. It is converted into mevalonate. And the enzyme is reductase. Right. What is the use of statins? Statins are going to inhibit reductase. जैसे if we talk about lovastatin is there, compactin is there, right? Atorvastatin is there. So in general, what is the mechanism of action of these statins? They are simply acting as competitive inhibitors of HMG-CoA reductase, right? This is its therapeutic use. Second example we can quote of the anti-cancerous drugs. The most important one is methotrexate. Now see, if we talk about methotrexate, it is again inhibitor of which enzyme? Folate reductase inhibitor, or you can say, I'll just simply write dihydrofolate reductase is actually the name of the enzyme, but we can simply write folate reductase inhibitor. And in my previous video also, I had described about the use of folic acid, and I used one very important term, 1-carbon metabolism. Folic acid is required for generating, for putting 1 carbon. So basically what happens here is simple reaction is there, we can say that uracil, which is the base present in RNA, is converted into thymine, which is the base present in DNA. So, if we see the structure, the most important structural difference between uracil and thymine is of the methyl group. And who's the donor of this methyl group? This methyl group is donated by this folic acid or folate. This is a simple reaction. Methotrexate is an anti-cancerous drug because when methotrexate is given, it is going to inhibit this reaction. So, obviously the concentration of thymine will decrease, or you can say it won't be synthesized, and the virus who has taken the host machinery will be unable to survive. And that is why we always say whenever a person is on methotrexate, folic acid supplementation should be given. So, I'll just mention it that folate supplementation is provided when a patient is on methotrexate. Because folic acid is required for other important things, and methotrexate, when a person is taking it, is inhibiting this reaction to occur. So, these two are the very, very important examples of competitive inhibition. And other examples we can quote that. Third example can be dicumirol. Dicumirol is actually an inhibitor of epoxide reductase. Epoxide reductase, which is again a very important component of this vitamin K. So, these three are the therapeutic uses of competitive inhibition. We can say that statins are there, competitive inhibitor of HMG-CoA reductase, methotrexate, folate reductase inhibitor, dicumirol, epoxide reductase inhibitor. Okay, other examples are also there. Allopurinol is also there. But why I am not discussing allopurinol here? Because allopurinol, if we see pharmacological action of allopurinol, no doubt it comes under competitive inhibition. But biochemically, if we see, it leads to suicide inhibition. So, even I can just mention it here that allopurinol, which I will be discussing in detail in suicide inhibition also. So, I'll just write allopurinol is competitive inhibitor of xanthine oxidase. Xanthine oxidase, and it is generally given to the patients who are suffering from gouty arthritis, okay, which will be explained later in detail in suicide inhibition. So, I can write xanthine oxidase, or you can say uric acid, or whenever there is hyperuricemia. Okay, so these are the very, very important examples, and the most important point which we have to remember in competitive inhibition, that Km will always increase because the affinity of the enzyme with the substrate will decrease, but Vmax will be unchanged. And competitive inhibition is strictly reversible. Why? Because if we increase the concentration of the substrate, it won't let inhibitor to bind with it. So, we can simply say that if you look at this, this is the equation. If the concentration of substrate increases, inhibitor will no longer be able to bind with the enzyme. That is why it becomes reversible. So, this is competitive or reversible inhibition, where we can say X-shaped graph is obtained when we talk about the Lineweaver-Burki's plot. Then the second type of inhibition is non-competitive inhibition. Now see, this non-competitive inhibition is actually, that is why I am not writing whether it is reversible or irreversible, because it can be reversible also, but most commonly it is irreversible. And we know the concept, we know the difference between reversible and irreversible. Reversible which can be reversed back, irreversible which cannot be reversed back. Okay. So, non-competitive is actually generally, I would say, most commonly irreversible inhibition. And I'll tell you the reason why it is like that, most commonly irreversible. Now, if we talk about, first of all, we will just try to understand the equation behind this. And if I write the equation, you saw in competitive inhibition, the substrate was combining with the enzyme forming enzyme substrate complex. Now what happens in non-competitive, it is the wish of the inhibitor. Inhibitor की wish है whether it is going and binding with the enzyme forming enzyme inhibitor complex, or it can go and bind with this forming enzyme substrate inhibitor complex. So these can be the two situations. Now what happens here is, if the situation is like this, once the enzyme substrate site is blocked, everything is blocked. So I can say that when enzyme substrate inhibition या inhibitor complex is produced, it is irreversible. Okay, and when it is binding with only the enzyme free inhibitor is binding, it can be reversible. But most commonly, as I mentioned it here also, it has to be irreversible. Now, if we talk about the kinetics, again there are two factors. One is your Km, other is your Vmax. Ab in competitive, the Km increases, Vmax same. Here what happens is, here nothing will happen to Km. So I can say Km will be unchanged, and obviously Vmax has to decrease. So I'll just highlight this. I can say that Km will be unchanged, whereas Vmax will decrease. And that is what exactly happens in non-competitive inhibition. And if I have to plot it as per Michaelis-Menten graph, we can simply say that x is y axis. So I can simply say that Km will be unchanged. This like this. So there will be a change in Vmax, whereas Km remains the same. And if we plot a, you can say line Weaver-Burkes plot, I can plot it here. So if we plot it like this, now here, since Km is same as I have mentioned, but Vmax will decrease, so I can simply say that it will be like this. And again the same parameters, this will be 1 upon V0 and this will be 1 upon the substrate concentration. So I can say that here again there will be a decrease in Vmax and this becomes Km. Or I can say this is V-shaped. So just to differentiate between this, I can say that V-shaped graph is obtained in non-competitive inhibition and X-shaped graph is obtained in competitive inhibition. Okay. Then moving on to what are the different examples. Now, if we talk about non-competitive, the most important example is fluoride. Fluoride ions are the inhibitors of enolase. Very, very important. This is actually a very important question. Fluoride ions are the inhibitors of enolase, which catalyzes conversion of 2-PGA to PEP. So since fluoride ions are inhibiting this enolase, whenever we have to estimate glucose in the serum, whenever a technician comes and take our blood sample, and whenever he comes, while withdrawing the sample, he will always ask what parameters have to be estimated in the blood. So if we say sugar, yes, if a person is suffering from diabetes, he will always go for glucose estimation. So he is going to take that gray vacutainer. So what is the purpose behind that gray vacutainer? Say this is the vacutainer. In this vacutainer, an anticoagulant is added, along with this sodium fluoride is added. And what is the purpose behind this? Sodium fluoride is going to stop glycolysis. because once he withdraws the sample, then he has to take the sample for processing to the lab. Okay. So always remember fluoride, it cannot be reversible. It has to be irreversible because whatever amount of glucose is there in the blood, it has come in the serum and it will be estimated by quantitative method. So this is one of the very ideal example of non-competitive inhibition, fluoride being an inhibitor of enolase, just to stop glycolysis so that correct estimation of glucose is done. And second example we can quote about cyanide. We know that cyanide, we've been studying this for donkey's years, that cyanide is a irreversible inhibitor of cytochrome A1, A3 oxidase. Third example can be fluoroacetate. Right? Fluoroacetate resembles oxaloacetate in structure, so fluorocitrate will be produced. So again it is going to affect TCA cycle. So these are another example of this irreversible, or you can say non-competitive inhibition, which is most commonly irreversible. Then after this there is different kind of inhibition. We can call it as a kind of sort of mixed inhibition. It is uncompetitive, which is very rare also. So the third one is uncompetitive. Generally it is a very rare inhibition. And there is only one example of this uncompetitive inhibition. Regarding the status of this, we cannot say it cannot be reversible also. Okay. So if we talk about this, the most important, first of all, let me just write the equation. The most important point which you guys have to remember is that the inhibitor will always bind with the enzyme substrate complex. This is one very important point related to uncompetitive inhibition. Because if you see in competitive, competition is there, so inhibitor is binding with the enzyme. If we see non-competitive inhibition, it is the wish of the inhibitor, whether it is binding with the enzyme or whether it is binding with enzyme substrate complex. But in uncompetitive inhibition, most specifically, in fact, this is the only reason that the inhibitor is going to bind only with the enzyme substrate inhibitor complex. Now coming over to the kinetics, what will happen? If we talk about, again we are dealing with these two parameters only. So Km and Vmax both will decrease. So I can say Km lowered and Vmax also decreases. This is uncompetitive inhibition, and again if we have to extrapolate it on the graph, so we can simply say that this is, say, Michaelis Menten, right? And this becomes, and you can see this graph is also a weird kind of graph. And if I have to extrapolate like this, it is like totally parallel. Like this, without touching the parameters, like this. And the most important point here to remember is, what is the example? The example only here is that placental, or you can say phenylalanine hydroxylase inhibiting phenylalanine in the placenta. So this is the only example of uncompetitive inhibition. So if we have to just totally, correlate all the different type of inhibition which we studied, I'll just quickly summarize. We can simply say that we studied three type of inhibition. One, we studied the competitive one. Second, we studied the non-competitive one. And third, we studied the uncompetitive one. Right. And if we talk about the differences, there are only two parameters, Km and Vmax, by which we can differentiate these two inhibitions. So if I talk about competitive, we know that now you also should be able to answer this. Km, competition, Km will increase. Vmax, same. Non-competitive, Km, same. Vmax decreases, and in uncompetitive, both decreases. Right. So this is just a comparative table between competitive, non-competitive and uncompetitive. Then after this inhibition, we come over to a very interesting inhibition, which is actually altogether a different type of inhibition. We call it as suicide inhibition. Now, as the name suggests, suicide. Matlab, once a person commits suicide, nothing is left. Same is the case if a suicidal inhibitor is there. If it comes, the reaction no longer proceeds. And there are only two examples in our body which are actually ideal for suicide inhibition. First of all, just try to understand what is this. So, what happens in suicide inhibition? I'll just write the definition. The inhibitor binds to the enzyme, right, and makes the substrate less potent. And the inhibitor, when it is binding to the enzyme, substrate ki potency it will decrease, and the complex will be so much powerful that it will not let the substrate to react with this. Okay, that is why we say the body has actually committed a suicide. For example, the ideal example of suicide inhibition is allopurinol. And allopurinol, actually, if we talk about the pharmacological aspect, the mechanism of action of allopurinol is it is a competitive inhibitor of xanthine oxidase. But in biochemistry, what we say is allopurinol, whenever it is taken, because it is a drug of choice to cure patients suffering from gouty arthritis. The reason behind that is if we summarize the reaction, what is happening actually, this is given in hyperuricemia. We know what is uric acid. Uric acid is coming from the degradation of purines. Degradation of purines. So if we draw purines, we know that AMP is there, GMP is there, HMP is there, but again I am not going into that much detail. I'll just focus on the major reaction. What happens is hypoxanthine is produced, whether it is adenosine monophosphate, guanosine monophosphate or inosine monophosphate, they are going to form hypoxanthine. This hypoxanthine will be converted to xanthine, and this xanthine will be converted into uric acid. This is the normal process which is going on. And the enzyme here is xanthine oxidase. And here also the enzyme is xanthine oxidase. Right. And obviously, if there is increased degradation of purines, there will be increased concentration of uric acid, leading to again inflammation and pain in the joints, leading to gouty arthritis. Now, when this allopurinol is given, basically it's a drug of choice. When this is given, it is going to block here or it is going to block here. So what happens is it is going to form allohypoxanthine here, or it is going to form alloxanthine. Now the question is, when it forms either allohypoxanthine or alloxanthine, they are actually both water soluble. So what will happen is, since they are water soluble, so they are easily excreted via urine. That is why it is drug of choice, because what happens is allohypoxanthine, alloxanthine, whatever is there, it will simply block the activity of xanthine oxidase, and the person can stop using it or stop taking it when the concentration of uric acid comes back to normal. So it tends to bring the uric acid level down. So why it is suicide inhibition? Because allohypoxanthine and alloxanthine, they are much more potent, they are much more stronger than hypoxanthine and xanthine. So that is why we say the body has actually committed a suicide. Halki it is beneficial for the body, because obviously uric acid has to be decreased, it has to be in the normal range. Okay. So, but generally we say that theoretically we would say that pharmacological or mechanism of action is simply competitive inhibition. But if we talk about biochemical action, it has to be suicide inhibition. This is one very important example. Okay, and I will write it down here also, that these forms are more potent, more stronger than the original form. So this is one very important example of suicide inhibition, and second example of suicide inhibition is aspirin. Aspirin, which is again a very frequent painkiller. If taken in high doses also, it is harmful. So aspirin ka, what is the function of aspirin? It is a suicide inhibitor of cyclooxygenase, or commonly we call them as COX. All the niacids, they are the inhibitors of COX. This we know that COX, which is again a rate limiting enzyme of in prostaglandin synthesis. So this is suicide inhibition. So till now what we have discussed competitive inhibition, we have discussed non-competitive, uncompetitive and suicide inhibition. Then the last inhibition, which is actually I would say the pillar of biochemistry, is allosteric inhibition. So which is actually known as modulation, but yes, if we see the function, we call it as inhibition. The next one is allosteric inhibition. So I will just use two words. First of all, allosteric modulation, oblique inhibition. So basically, what is the difference, which we have to actually understand. Now, basically this allosteric, it is derived from two words, allos and steric. Allos means other and steric is the interactions. Right. So what happens is, say this is the enzyme and this is the substrate. Okay. And say here lies your modulator. This is your modulator. I'll just name it as A. So what happens is, this allosteric modulator can ping this enzyme from anywhere where he wishes, from here, from anywhere. So I can say that the allosteric modulator is going to attack any other region in the enzyme molecule irrespective of the active site. It is not going to disturb the active site. It will never disturb the active site. But yes, it might be possible that since enzyme is a protein, it might go and bind somewhere because it's a three-dimensional structure. But whatever will happen, since it is a protein, there will be a change in the interactions. So if those interactions are, there can be only two reasons. One can be, it can be favorable, or other can be, it is harmful for the enzyme. There can be two possibilities only, ya to it will be harmful, ya it will be harmless, or it will help. So whenever it is helping the enzyme, we call it as a positive cooperativity. And when it is not helping, we call it as a negative cooperativity. But it is not cooperating. But this negative cooperativity can be taken as a kind of help. And this negative cooperativity is generally referred to as feedback inhibition. Very, very important. Right. And but if it is helping, it is positive. And the most important, ideal example of positive cooperativity is binding of oxygen to heme. We all know that when one molecule of oxygen binds, okay, it will call other friends also to bind with heme, 1, 2, 3, 4. So it is a positive cooperative binding. And if we talk about negative, there are so many reactions of negative cooperativity. Just suppose if you are seeing my video, if you are watching my videos, if you like these videos, obviously you are going to give a good feedback. And if it is negative, so that means you are not liking, so it will be a negative, so all other students might not also watch. But if you give a positive feedback, automatically it will be there. And we all know that negative feedback, positive feedback, this all you have covered in physiology, which you have actually studied in detail. So there has to be a positive feedback also, there has to be a negative feedback also. So negative feedback, I am just saying कि इसमें क्या हो जाएगा, it will be known as a inhibition. Positive feedback will activate a molecule. Okay, and positive cooperativity is altogether different. But overall, it will all come under this allosteric modulation. That is why I told you allosteric inhibition, basically it is a misnomer. So we should call it as a allosteric modulation. And if we have to define this, I can simply say that allosteric modulation is defined as that type of inhibition in which the modulator goes and binds to an area totally different from the active site, somewhere around the enzyme, thereby altering the enzyme substrate complex, thereby altering the conformations. If it is helping, it becomes positive. If it is not helping, it becomes negative. And the most important feature about this is that the most important one is what kind of graph will be obtained, because whenever we are talking about a mathematical calculation, we have to be actually thinking about the graph. So if I talk about the graph, I can simply say that in Michaelis Menten equation, this was the graph, saturation kinetics. Allosteric mate is not possible. Why? Whether it will be positive, whether it will be negative. Okay, it cannot be a hyperbola, so I can simply erase this graph. Right? So what kind of graph is obtained? This is sigmoidal curve. So always remember that in allosteric modulation, sigmoidal curve will be obtained. Now, if sigmoidal curve is obtained, it is different from Michaelis-Menten equation, which gives a hyperbola. So, the main difference, we can simply say that if it is giving a different curve, I can say that allosteric inhibition या modulation never follows, very important, never follows Michaelis-Menten kinetics. Kinetics, this is very important. So this one has to keep in mind that this is the difference, that so it is always different. So there is no concept of Km and Vmax. But yes, there were some scientists who tried classifying them into Km and Vmax, but again it will never follow this Michaelis-Menten kinetics. So we can say that there will be separate allosteric modulators, there will be separate allosteric inhibitors. So again, now we will go over the example. So if we talk about the examples, I'll just give you one very common equation which all of you must be knowing, that is fructose 6-phosphate is converted into fructose 1,6-bisphosphate. First of all, it should always be bis, because most of the students get confused between bi, di. It cannot be bi, it cannot be di, it is always bis. Bis means adjacent. As you can see, one is present on the first position, another is present on the sixth position. And the enzyme is PFK1, again a rate committed enzyme or rate limiting enzyme also of glycolysis. Here, if we remember that if we talk about the inhibitors, it is inhibited by citrate and it is inhibited by ATP. But it is activated by AMP. So this is allosteric modulation. So AMP is an allosteric activator of PFK, whereas citrate and ATP are inhibitors of PFK. This is one very important example. Then if we talk about the second example of this, again, pyruvate is converted into oxaloacetate. The name of the enzyme here is pyruvate carboxylase. Very important. Again, we can say ATP is required here because carboxylation is there. And the most important thing here we have to remember is acetyl coenzyme A is a allosteric modulator of this reaction. Because acetyl coenzyme A, this is a reaction of gluconeogenesis. I'll just brief the things to you because in any case we will be discussing these all reactions in detail. So what happens during gluconeogenesis? There is synthesis of glucose during fasting. So during fasting, the oxidation of fatty acid will also increase. So acetyl coenzyme A, poor chap, cannot be directly converted into fats. So this is an indirect way. It will promote gluconeogenesis so that our body can get glucose. Because just keep one thing in mind, our heart, brain, these are the two very, very important organs which just cannot stay without energy for even a parsec of second. And RBCs, so yes, because RBCs are the ones which are responsible for flowing off the blood along with nutrients from head to toe. Okay, and that is why we say RBCs, no matter what is the state of the body, whether we are sleeping, whether we are eating, whether we are doing any damn thing, they always depend on glucose for energy. And that is why we always say this also, diabetes mellitus is the one which is affecting the RBCs, thereby affecting all organs side by side. Okay. So these two are the examples of allosteric modulation, and the most important point which you have to remember it never follows Michaelis-Menten kinetics, and second is they always give a sigmoidal kind of curve. Now, just now I told you that it is an example of feedback inhibition also. So what is this feedback inhibition? It is simply inhibition by the product to this enzyme. So whenever a product starts increasing in concentration, it starts inhibiting its own production. This is a simple universal law also, which is also known as product inhibition, which is also known as allosteric inhibition, like this, right? So I can simply say that feedback inhibition mein there are numerous examples. For example, glucose to glucose 6-phosphate. This is feedback inhibition. If I talk about, so I can say the enzyme here is hexokinase, and if there is increased concentration of glucose 6-phosphate, it will inhibit hexokinase. Similarly, if we talk about heme synthesis, succinyl coenzyme A plus glycine catalyzes the synthesis of heme, or I would just write the main one, ALA, and the enzyme here is ALA synthase. And this ALA by different reactions form heme. So whenever the concentration, whenever this heme increases, it can only give instructions to ALA synthase. Otherwise, if we look at heme synthesis also, there are so many steps. Okay, but it can give instructions only to the rate limiting enzyme. So this is again very important fact to understand, that feedback inhibition is mainly restricted to the rate limiting enzymes or rate committed enzymes, or overall irreversible steps or enzymes. Because reversible hai, to there is no question of inhibition, because reversible ka kya hai? So वैसे it will be converted back to the substrate sooner or later. Okay, so this is allosteric inhibition. So these are the very, very important type of inhibitions which can be categorized into reversible, which can be categorized into irreversible. Then after inhibition, and again the most important one is, we should also not forget the kinetics. Kinetics is very important to understand. Then after this inhibition, then we are going to touch another very important topic, regulation of enzyme activity, which is again very important one, regulation of enzyme activity. Now there are so many ways by which we can actually regulate this enzyme activity, but we would be concentrating on mainly two parameters. One is allosteric modulation and second is covalent modification. So if we talk about allosteric modulation, I will simply write feedback inhibition also. So because just now I discussed it, so it will be easier for you people to understand. Okay, so if I have to explain this allosteric modulation, one more reaction I can explain, which is again a very ideal example of this allosteric modulator, that is in urea cycle. If we talk about the first step of urea cycle, where what happens is these bicarbonate ions combine with ammonium ions to form carbamoyl phosphate. Here the enzyme is CPS1, which we will be discussing obviously in detail. It stands for carbamoyl phosphate synthetase 1. So even if you don't remember the enzyme at this moment, we will discuss this enzyme later when we will discuss urea cycle. Here I just want to tell you how allosteric modulation is actually governing the entire process. So here what happens is, here obviously ATP will be there, 2 ATP converted to 2 ADP plus 2 inorganic phosphate. The most important thing is positive sign. Wherever you see positive sign, okay, it is always activating. Wherever you see negative sign, it is always inhibiting. So here you will find this term NAG, which stands for N-acetyl glutamate, whose function is actually to bring more and more of ammonia, because urea cycle, urea is the most important, I would say, the excretory product in human beings, which comes from the degradation of the proteins. And if any kind, if there is any factor which is related to increased concentration of ammonia, it will again simply affect the brain the most, which again we will be discussing, which we commonly call it as ammonia toxicity. Okay, so this ammonia needs to be removed, and how it is removed from the brain? It comes from glutamate forming glutamine, and ultimately it has to hand over everything to the liver so that urea formation takes place. Okay, so that is why it is an allosteric modulator. It is helping in bringing more and more of ammonia to get detoxified. Okay, then we come over to allosteric. We have discussed already in detail, so I am not going to touch it more. Now we are going to study this covalent modification, which is the basis of the metabolism in biochemistry. So if we talk about this covalent modification, what is happening? See, we can simply say that modification of something in a covalent way. Okay, so if we talk about any regulatory enzymes, they will be present in two forms. One is active, one is inactive. Just say, for example, suppose if we are entering in a dark room, obviously we are going to turn on the lights. When we are going out, we are turning off the lights. Similarly, whatever is required by the body, that will be automatically switched on. If it is not required by the body, it will be switched off. So that on and off is nothing but the modification in a covalent way. Okay, and the most important ones which we have to remember is this covalent modification can be reversible, it can be irreversible also. It can be irreversible, it can be reversible. First of all, just try to understand the concept. What is this covalent? Covalent means we know covalent bond is considered to be the strongest bond. So what happens is there has to be a change in between the forms. Jaise active will be converted into inactive and vice versa by modifying it covalently. Irreversible means वो वापस नहीं हो सकता है. Reversible means it can be again reversed. So if we talk about irreversible, we can simply quote the example of zymogens or proteolysis. Once the enzyme is active, it is going to degrade. No matter what happens, it cannot be reversed back. क्योंकि digestion cannot be reversed back. ना जब हम लोगों ने when we have taken food inside, so automatically it cannot go out in a normal individual. Okay. So zymogens will be under irreversible ones. कि when the reaction जैसे chymotrypsinogen has acted, it cannot be converted into the prochimotrypsinogen back. But if we talk about reversible में there are so many examples. जैसे the most important example is phosphorylation, acetylation, methylation, and ubiquitation or ubiquitin addition. One more is there, ADP ribosylation. So these are the 1, 2, 3, 4, 5 more important, the most important reversible type of covalent modification. Irreversible, I have told you, it cannot be reversed back. But yes, reversible means it can be reversed. So if I talk about phosphorylation, dephosphorylation would be the term. If I talk about acetylation, deacetylation would be the term. Methylation, demethylation, ADP ribosylation, and ubiquitin, again they will be reversed back. Okay. Now if we talk about phosphorylation, the most important one which is actually used in biochemistry. So I can say that phosphorylation and dephosphorylation. Okay. Now see what is happening here is, first of all we will take the example of two enzymes. Okay. I will take two processes basically. One process is glycogenesis. Genesis, wherever you will find the term genesis, it means synthesis of glycogen. Second is glycogenolysis. Lysis means breakdown of glycogen. Okay. So if we talk about glycogenesis, no doubt we are going to cover these again processes in detail, but I'm just telling you, just in our body there is a well-fed state when when we are having a high carbohydrate diet, whatever amount of glucose we are taking, it will be converted and stored in the form of glycogen in the liver as well as the muscle. So here the enzyme is glycogen synthase because it is synthesizing glycogen. So glycogen synthase. And if we talk about lysis, the enzyme is glycogen phosphorylase. Right. So this glycogen synthase is present in two forms. Similarly, this glycogen phosphorylase is also present in two forms. One is the active form, the other is the inactive form. Same active and inactive. Right. So I can say active can be converted into inactive or vice versa. Similarly, active can be converted into inactive and vice versa. So what kind of enzyme is this glycogen synthase? It is having this type of structure where serine is present at the active site. So I can say same is the structure here. Now what happens is that HH is there. This H can be, because this is a covalent bond. How it will be modified? We can simply say that this H will be phosphorylated. So I can say if only HH is there, we can't find any phosphate here. So this is dephosphorylated. But if phosphate comes here, PP, it becomes phosphorylated. So I can simply say that there can be a interconversion of dephosphorylated to phosphorylated and vice versa. So if a phosphate is attached, always remember there is the enzyme is kinase, which we will be discussing in classification. So here I'll just write kinase. Means it is attaching this phosphate. But if phosphate has to be removed, the enzyme is phosphatase. So it will remove phosphate. So I can simply say that there is a there is a covalent modification, which means that dephosphorylated can be converted into phosphorylated and vice versa. Okay, so if we talk about whenever there is more concentration of glucose, automatically synthesis of glucose, synthesis of glycogen would be more. So I can say increased glycogen synthase, or the active form of glycogen synthase will be there. And just for your knowledge, till here I can simply say that active form is dephosphorylated. We are going to discuss this again in the metabolism of glycogen. Okay. Similarly, you can say that when concentration of glucose is more, glycogen synthesis will be more and the active form will be dephosphorylated. On the other hand, when there is less glucose, body will be obviously triggered to form glucose. That means glycogenolysis will be more. So glycogen phosphorylase will be made active. So glycogen phosphorylase active form needs to be phosphorylated. So you can say that there will be a change in the phosphorylation and dephosphorylation status depending upon the activity in the body, right? So this is one example. I can say that glycogen phosphorylase and synthase. Another example can be PDH, pyruvate dehydrogenase, which is again present in active and inactive form. So its active form is also dephosphorylated. So inactive form has to be phosphorylated. Similarly, another example is HMG-CoA reductase. Again, same active form will be dephosphorylated and inactive form will be phosphorylated. So right now, I think this much is enough. So slowly when we are going to study the reactions, this concept will be automatically clear to you. So this is nothing but covalent modification, because whenever our body is, just suppose if we go in a hypoglycemia, immediately cyclic AMP will trigger glucagon, which will be released from alpha cells of pancreas to initiate the process of glucose formation by increasing glycogenolysis, by promoting glycogen phosphorylase. So this phosphorylation is there. Acetylation or deacetylation is there, which is required in regulation of gene expression, which we will be discussing in detail in MOL Bio. If we talk about methylation, again demethylation, which we will be discussing in MOL Bio again, where we are going to study the epigenetics. Ubiquitin is again simple, it will lead to degradation, and ADP ribosylation, which again in the molecular biology, which has a very important role to play into cholera toxin. So this is a very important covalent modification, and this is how the regulation of enzyme activity is done. So this is all about in this video. Then in our next video, which will be the last video on enzymes, we would be discussing about the classification and what are the clinically important enzymes. Thank you so much.